Well, thank you very much, and it's good to be here at BASH and to bring the Technology That Matters event here. And we do this in partnership with both SGInnovate as well as A-Star Central and with MTech Asia. This is the fifth Technology That Matters event that we're doing. You know, we do it on a monthly basis. I see several similar faces who have been to the Technology That Matters in the past, and welcome again. For those of you who are here for the first time, this is an opportunity to hear really a thought leader speak, and then to have a fun cocktail party afterwards where we can all share ideas, because in many ways, you're actually the stars of the event, coming here and sharing with each other and networking. But it's with great honor that we have a professor from MIT, Peter Dedon, here, who's an expert in genomics, and today he's going to be sharing about epi-everything. So the convergence of technologies that yield new opportunities beyond the genome. After he speaks, then Daria will be telling us a little bit about an exciting competition that we have, Innovators Under 35, and then after that we'll be having our cocktail party. So let me tell you a little bit about Peter as an introduction. He has his MD, PhD training in cancer biology. Peter's research program applies chemical approaches to understanding nucleic acid biology, and he'll be talking about that this evening. He's the Underwood-Prescott professor in the Department of Biological Engineering at MIT, and also at SMART, Singapore-MIT Alliance. He's the principal investigator in the Antimicrobial Drug Resistant Program. The thing here of his credentials as an academic is huge, so I'm not going to go through all those, but just say that from a entrepreneurial perspective, the research that he's doing both at MIT and here is developing new enzymatic tools for biotechnology, new methods for industrial microbiology and protein production, and novel antimicrobial agents for screening and structural-based drugs for programs as well. And so it's really an honor to have Peter here. So if we could put our hands together and welcome him to the stage.
All right. So thank you, Howard. I won't do a prolonged introduction. I think some of you may have heard a talk that I gave at the MTech Asia event several months ago. And what I've done here now is to add some techno meat to this, because there's fewer rocket scientists and more biologists in the group. And what I want to do is really talk about some technologies that have made a huge difference in how we do biomedical research in terms of opening up a new frontier, if you will, in genetics. And it's beyond genetics. This is all about the epigenome and the epitranscriptome, which are fairly new areas. Epigenetics is maybe a decade old in terms of the kinds of advancements in understanding how it works. Epitranscriptomics, the word was only coined a couple of years ago in terms of what we want to do. All right, so this does require a little genetics 101. And I am going to move through this pretty fast, but we go back in genetics all the way to the discovery of DNA and the structure of DNA, and the work of Rosalind Franklin that was stolen by Wilkins and given to Watson and Crick in that horrible story that nobody likes to remember. They get the credit for discovering the structure of DNA. A meter-long polymer made up of 5 billion nucleotides of repeating units of A, G, C, and T in a double-stranded helix, containing all the genes necessary for coding for a human being. It's about 750 megabytes of information in there. That's all you need to make a human. What's interesting is the touchiness of the buttons. Where this works is this meter-long piece of DNA gets broken up into 46 chromosomes in every cell, 5 billion nucleotides of the DNA, about 2.5 billion base pairs. We know now that there are about 20,000 genes, and we'll look at that in a minute. 23 chromosomes from mom, 23 chromosomes from dad. Everything you need to build a human is there. except for a couple of pieces. So the oldest way to explain how does the genes in DNA get turned into proteins that then become the functional units of a living organism, it's the central dogma, the classic central dogma of biology. Every high school student learns it. DNA gets transcribed into RNA. The RNA then gets translated into protein. Proteins are the building blocks that make the cell function, and there you go. All the genes get turned into their functional units. So the most important element here is the genetic code in the DNA. So genes are comprised of units that we'll call codons. These are 3 nucleotide repeats. Each different 3 nucleotide combination specifies an amino acid. So there's the connection between the genetic code and the proteins. Now, what we've got is a situation where you've got all possible combinations of 3 nucleotides gives you 64 codons. We only have 20 amino acids that make up proteins in cells, 21 if you count selenocysteine. So we have more codons than we have amino acids, and that's important when we talk about epitranscriptomics and epigenetics. So there's our genetic code. The whole concept of transcription, a little cartoon to show the double-stranded helix. It's going to get separated here, and along comes a polymerase and copies the information in the DNA, turns it into RNA, and the RNA, the messenger RNA, and all the other RNAs peel off and head out to do their function. So the whole collection of all the transcribed RNAs, not just messenger RNAs, but all the structural and functional RNAs, is the transcriptome. And what we're concerned about today is really messenger RNAs and tRNAs. Those will be the actors that we talk about in terms of the epitranscriptome. So the messenger RNA heads out to the cytosol, goes to the ribosome, and while it's there, these triplet codons get read by the tRNAs. So the tRNAs read the codons with their anticodon, and they have the amino acid attached to them. So we get this continuous process of synthesizing proteins by reading the messenger RNA like a ticker tape. That's the central dogma at work in terms of the translation. So DNA gets made into RNA, gets made into proteins, has been ruling the world of biology for decades. But the problem was we still didn't know how many genes we actually had in the human genome up until just a few years ago. And this is where the importance of the Human Genome Project came along. There's a convergent technology which allowed us to sequence every single nucleotide of the 5 billion and get them in the right order. Turns out now that there are about 20,000 genes in the human genome, right? That's a pretty small parts list to create a human being. So how now, we're in the post-genomic era, if you haven't heard that. Genomes have been sequenced. You can get genomes sequenced for a couple of $1,000s now, entire genomes, every single nucleotide. It's easy, it's done, it's no longer a big scientific experiment. So we're in the post-genomic era. What do we do with all that information now? Well, one of the problems is how do we assemble 20,000 genes into a human? We still don't know. We know the parts list, but we don't know how to do the assembly. You can look at the human genome like a box of Legos, 20,000 Legos, all right? And the assignment is to build a replica of the city of New York, which is about as complicated as a human body, from those 20,000 parts. Well, we have a problem. We know the parts that are there, but we don't know where to start. We don't know the order in which to put them. So how do we go from a human fertilized egg to an adult with the development of all the 27 organs in the body, trillions of cells, with 20,000 parts without an instruction manual? And this is where the epigenome and the epitranscriptome come in. The epigenome and the epitranscriptome are the schedulers of gene expression. They tell you when to do it and how much to do so that the timing is right. Turning on the 20,000, turning off the 20,000 when we need them and where we need them, so that we can go from a fertilized embryo through organogenesis and development into a full-fledged human being. Very complicated. Some of us have hair, some of us don't. All of those things are controlled by the epigenome and the epitranscriptome. What they really consist of, what the epigenome and epitranscriptome really consist of, are hundreds of different chemical modifications of DNA and RNA and proteins. It's information that's added to the DNA, that's added to the RNA, that's added to the protein after these things are made. So it's epi. It's above, outside the genome. We do not change the genetic code. We simply tell the genes when to turn on and when to turn off. So the epi allows us to transcend that. It's a user manual. It does schedule. Start at part A, put part A onto part B, add 4 parts of part C to get us that model of New York City out of a box of Legos. But this is 20,000 genes. Now, this is a little silly, right? We have 2 identical twins, but the point is they have identical genomes, exact same DNA sequence, exact same genes. But everybody knows that identical twins are still different. One could get a disease, the other might not. They age differently, they have different traits and different qualities. Part of this is thought to be due to variations in the epigenome and epitranscriptome in the scheduling of gene expression. It varies slightly, even among identical twins. So here we are, this magnificent vista looking out at some grand horizon for the epigenome and the epitranscriptome. How does it work? And this is where I want to talk today about technology that matters, because there's a lot of new technologies that have allowed us to see into the epigenome and the epitranscriptome in ways we couldn't have done 10 years ago without these big systems biology level technologies. Now, I said previously that the epigenome and epitranscriptome, even the epiproteome, are nothing more than chemical modifications of the polymers. We know that there are over 200 different chemical structures that get added to DNA, RNA, and proteins to alter their function and to alter gene expression. A huge information system. And we'll look at the information content as being a very important part of technology that matters in terms of deciphering this. How does it all work? There's going to be a quiz on all the structures here during dinner. No wine or beer until you can identify all 150 RNA modifications. Okay, how does it work in DNA? There's 4 or 5 different chemical modifications of DNA. 5-methyl-C is the one everybody learns in school. They get put on different genes at different times in different locations. The DNA in the nucleus is wrapped around 8 proteins in a nucleosome, and these proteins are pairs of histone proteins. They have tails. Those tails get decorated with an epigenetic code of 30 different chemical modifications. So the combinations of histone decorations, DNA decorations, define when a gene gets turned on, turned off, when silent chromatin becomes active chromatin in a code. There's a code there. We just don't know what it is yet. The simplest way to think about the epigenetics is where we have DNA, and the gray circles are like 5-methylcytidine. And any gene that has 5-methyl-C on it is not being transcribed by the red Pac-Man RNA polymerase, but the genes that don't have 5-methyl-C get transcribed. There's our scheduling. When you want to shut a gene off, put 5-methyl-C up and down the sequence. That's a simplification, an oversimplification of how it works, but it's generally what we know at this point in terms of a simple model. Okay, RNA does the same thing. So messenger RNA, tRNA, every form of transcribed RNA we now know gets chemically modified with one of these different chemical structures. So another simplified model that's out there is that the messenger RNA that gets modifications on it is somehow stabilized against degradation, but any messages that don't have those modifications get sent to the trash heap, and that's a way we can schedule translation by controlling the stability of a messenger RNA molecule. Again, very simplified just to make the point. So here we have a genetic code, 20,000 genes, codons, very complicated. But now we're saying we have 200 different chemical modifications that add information to that in terms of when to schedule. Well, there's an epigenetic code. There's a group out there, a consortium out there trying to correlate behavior of gene expression with all the modifications on the histones, all the modifications on the DNA. It's a huge project. There's an epitranscriptomic code that is even less well defined, but that's what my group does and a lot of other groups, figuring out how do these 50 different chemical modifications in every cell turn on and turn off translation. So not to be too silly, but this is getting to be like a Dan Brown novel, all right? Codes within codes within codes. And we're all going to be Tom Hanks and figure out how to crack the code. It really is, how do we solve these codes, solve the riddles? And if we can do that, then we can understand function. If we understand function, we can now exploit it for drug development, for disease diagnosis, for biotechnology. So we have to be code breakers in the sense of trying to figure out how this works in order to get all of these good, useful outcomes. Go start a company that makes a new drug based on the epigenome or the epitranscriptome. All right, here's where technology that matters really matters. So about 4 or 5 years ago, a group of professors at MIT got together and wrote up a document, a white paper, on something they're calling convergence, convergent technologies. So the idea is that the first revolution in biomedical science was the ability to clone genes, right? The biotechnology revolution. Then was the human genome, the genome sequencing revolution. Sequencing all the genomes, developing that technology. Well, we've got that now. And the claim is that we're in a third revolution where we bring together life sciences, physical sciences, and engineering to develop technologies that allow us to get much more information out of biological systems and to do things that we simply couldn't do before. All right, so what I want to focus on is one form of convergent technology, systems biology. So typically, in the past, scientists, you want to measure something, you measure one molecule in the cell, and you develop a technology to measure that. Well, if we want to do proteins, we could do one at a time on a Western blot, but there's 20,000 of them. So we need a technology that measures all 20,000. All right, same thing with transcripts. All the messenger RNAs that are produced, thousands of them. How do we get them all in one fell swoop? How do we measure all the small molecule metabolites in a cell? That requires very sophisticated new technology and a combination of new technologies now. For example, if we want to do the measurements, we need sophisticated mass spectrometers. The new mass spectrometers are capable of measuring down to single molecule level and measuring all the molecules in the cell, all the proteins in the cell. We have the genome sequence for the human genome, and now to be able to use that information, coupling with computer science and informatics and writing algorithms that let us sort through trillions of bytes of data to find patterns, to get biological information out of an entire system. So now we can look at a cell or a tissue or even a whole human, take measurements, measure all the production of proteins, all the gene expression that's going on, and begin to define patterns and crack this code. All right, so the technology I want to talk about today is really focusing on systems biology. Convergent technologies are huge. Nanofabrication to make nanoparticles, layer by layer, particle technology to release chemicals out into biofilms to dissolve them so we can kill bacteria. That's an example of a convergence of technologies that's allowed us to make a breakthrough. We'll focus on systems biology here. And I want to use our recent discoveries in the epitranscriptome to illustrate how this all works. And what we discovered was that cells, when they get stressed, starve them of food, rob them of oxygen, treat them with a toxic chemical, that there's a program, there's a series of 3 systems that get activated to translate proteins when we need them and how much and where we need them. All right? And it all starts with transfer RNAs. We talked about those, the things that carry the little amino acid balls. And they have their anticodons here. There's 50 different types of tRNA in a cell, and they come in different copy numbers. There's 1 up to thousands of each one present in the cell. What we have discovered is that under stress, some of those tRNAs then get their modifications changed by enzymes, a series of enzymes, that alter 50 different chemical modifications in any one cell type. Those modified tRNAs then go and read specific messenger RNAs. And here's where the third system comes about in finding that genes are organized in the genome by virtue of using codons differently. And the key came when we discovered that the reprogrammed tRNA only selectively reads specific genes that have specific codons. Those genes turned out to be all the proteins you need to survive the stress that started all of this. So we've kind of slightly cracked an epitranscriptomic code in being able to identify tRNAs and modifications that allow us to schedule protein production when we need it in a crisis. So how do we do that? Okay, let's walk through sort of example by example here in terms of cracking this code.
It's based on the same core technology that's the foundation to
I don't want you to
thousands of
actually hear the words that
publications
this guy is saying.
very high quality sequence data that's been generated on
Listening to
Illumina's
me.
GA platform initially, and then the HiSeq platform.
All right. That guy was much more interesting than I am. what the point of this video was, this is a cartoon of next generation sequencing technology made by the Illumina Corporation. They don't mind if I pirate their product as long as I'm telling you about Illumina sequencers. Very sophisticated ways to do DNA sequencing and RNA sequencing. Now, we've used this a lot to be able to go quantify all the tRNAs expressed in a cell at one time. And what you get is this sort of New York City canyon plot looking down the rows and streets of New York City. It looks like skyscrapers. Well, each column here is a time point. There's 50 different tRNAs here. We're able to quantify them all at once and look at their behaviors as a system. And what you see is that under this is bacteria subjected to starvation. Here they go up a little bit and then they drop, but then when we give them food again, you get some skyrocketing tRNAs. We now know that those tRNAs match proteins that get super upregulated when the cell starts to grow again at that point. So we can look at the whole landscape of all the tRNAs and now study their behavior in ways we would measure only one at a time before. We miss all of this big systems level transcendent behavior. Okay, so those tRNAs get modified. We said that there's 50 modifications in every cell type. About 10% of the nucleotides in tRNA get modified chemically. How do we measure all those modifications at once? Again, we'll use the power of mass spectrometry to go quantify all of them. And what you see here is a picture, a chromatogram. Each peak represents a different chemically different building block of RNA. There's 40 of them that we're measuring in this one bacterium. We won't name them all. It's just to impress you with the quantity. Every tRNA's got about 8 of these different chemical modifications on them. There's 50 types of tRNA, 50 types of modifications. Think of the information content in the permutations and combinations in a way that controls how the tRNA reads its codon. Again, a huge systems level perspective on information that you wouldn't get by looking at any one modification alone. All right, now I want to show you, this is where I didn't show in MTech, is some real scientific nitty-gritty. Part of the power here is not just the measurement, but we also have to mine the data. And here we use multivariate statistics. This is a heat map that allows us to look at what things are going up together and what things are going down together. It's a statistical tool. And what we've got is that every row is a modification. There's 40 of them here. And every column is a time point after we rob the cells of oxygen. So when there's lots of oxygen, we got some modifications that are at high levels, some that are at low levels in the tRNA. But now you shut off the oxygen and they start to get, you're robbing them of oxygen, they're running out of oxygen. The cells are programmed to survive when there's no oxygen, and they're turning on a set of stress response proteins with this epitranscriptome and epigenome. And here, all of a sudden, the modifications that were high at this point have gone away, and there's other new ones that have come up. And you keep walking through the whole cycle of survival in hypoxia, and you get patterns. These patterns, if you just give me tRNA, I can tell you what chemical stress you were exposed to by simply looking at the relative amounts of modified nucleosides with about 85% accuracy. So there's a huge amount of information in these patterns, in this epitranscriptome. So how do we crack this code? There's a code here that's telling us how to survive hypoxia. Well, this is where now you have to start to use other convergent technologies. We're going to use genomics in a big way. Basically, what we know is that when we change modifications on tRNAs, it changes the way they read codons. And there's 50 different tRNAs for 50 different codons, 64 different codons. We can duplicate a few usages. The modifications reprogram them to read different codons. So let's go back to the basics. We've got 20 amino acid building blocks of proteins. The genetic code, 3 things taken one at a time. We can have 64 possible combinations. What we have is too many codons. We've got spare codons. We have several codons that will specify one amino acid. Those are synonymous codons. They translate to the same thing, but they're different chemical structures. That means we also have spare tRNAs. So These spare codons we discovered are not just randomly used. For years, people have had hypotheses about, well, why do some genes have more of one synonymous codon and others have another? Well, highly translated genes tend to use highly common codons. Well, we found another reason for this. When you take DNA out of a chromosome, stretch it out, you get a string of genes. The genes are all lined up. And I've got them in different colors here because we've discovered that different genes have different codon enrichments. And during a stress, we get selective translation of only certain genes. If we are exposed to radiation, your cells will translate into proteins only certain genes you need to survive radiation. Same thing with caffeine stress. You're shaking after caffeine. That's a cellular response to caffeine. We get certain genes translated there. Well, what we discovered was this cartoon here is not a cartoon. It's not a modern art, though you could frame it and it looks nice on the wall. This is the product of a sophisticated algorithm my colleague Tom Bagley developed to count all the codons and determine their frequency in all genes in the human genome, okay? Each row is one of 25,000 human genes. Each column is one of 64 codons. Here we have quantified all the codons in all the genes and do a multivariate statistical analysis so that everything that's similar is grouped together. Purple means this is a cluster of genes where these codons are all very significantly overused compared to the genome average. The yellows are underused codons. And by doing that, we identified, for example, families of radiation stress response genes that all share a very significantly overused synonymous codon. Amino acid is the same. We can change the codons and we can alter the translation efficiency, but it's all based on how the cells use different codons. So again, a third level of information, we've kind of cracked an alternative genetic code. Now, I got in trouble with some reviewers of a paper who said, how dare you use alternative genetic code? You're not that good. All right, call it auxiliary information in the genome. I don't care what you call it. But there's something there about codons that are used differently in different things. Now, what you end up getting when you put all this together, you got tRNAs that get reprogrammed with modification to read their codons. The codons for surviving radiation stress are enriched in messenger RNAs that we need to survive radiation stress. There's the epitranscriptome of the tRNA, slightly cracked. We now have an idea. I can go back. If you tell me all the proteins in a cell at any moment, I can go back and say which tRNAs are going to be modified and how many copy numbers of those tRNAs there be. And we've done that with great specificity. So we can understand the code now and what it means. All right, so why are you sitting here waiting for your wine and your food if I don't come up with something practical? Well, this is a big deal. So technology that matters really got us to where we now have discovered these big systems level things that you couldn't discover except by using these big technologies. The cool thing is, from a scientist's perspective, every living organism has DNA modifications, RNA modifications, protein modifications. Viruses, RNA viruses, have their genomes coded with modified nucleosides. Parasites, we're just publishing a paper in malaria, that the development of the malaria parasite depends on waves of tRNAs getting modified to control the expression of proteins you need to convert from a schisant to oh my, malaria friends would kill me, the first stage to the last stage of the parasite. Okay, so what are some applications? People who want to start companies, what can you do with this now? How can we make use of this kind of thing? Well, if you're a basic medical researcher, we now know that epigenetics contributes to virtually every human disease in some form. An error in a protein that puts the 5-methyl-C in the wrong place at the wrong time, the wrong histone code. There's a lot of people looking at cancer, especially, as a disease of epigenetic problems, all right? Epitranscriptomic errors, it's in its infancy, but one of the things that popped out from years of research is that there are many strange neurological diseases due to specific defects in enzymes that put modifications on the tRNA. We don't know how or why, and I would bet, I'll bet you a glass of wine, and if I lose, I don't have to pay anyway, but that, again, epitranscriptomic errors are going to be discovered. We're going to find single nucleotide polymorphisms in the enzymes for the hundreds of enzymes for RNA modifications in humans and bacteria and the like that alter the epitranscriptome and cause disease. Okay, in terms of drug development, there's opportunities here. What you're seeing here is an antibiotic being synthesized, being constructed at the active site of a tRNA modifying enzyme in a bacterium. It's a protein that puts a modification, a methyl group, on a specific tRNA. Turns out the protein is required by all pathogenic bacteria. So if we can make an inhibitor, we now have a beautiful antibiotic. So the epitranscriptome provides us with a huge number of potentially really good targets. Well, my favorite theme these days is antimicrobial resistance, because I lead a large antimicrobial resistance research group over in the CREATE buildings. And what I've got here is a cartoon where the epitranscriptome plays a huge role in drug resistance. There's an enzyme called erythromycin resistance methyltransferase, ERM. And it threw that purple ball, which is a methyl group, onto an adenosine, a nucleotide in RNA. Only one of about several thousand nucleotides, this enzyme modifies it. And by putting that simple methyl group there, the erythromycin antibiotic cannot bind to the ribosome and cannot kill the cell. So this is a huge form of drug resistance. Now imagine if we could make a small molecule inhibitor of ERM, prevent this chemical modification of the RNA, and allow erythromycin to go do its job. Erythromycin is cheap, safe, and easy. It's a great antibiotic. Well, let's make a resistance reversing therapy that goes after the epitranscriptome. So, not surprisingly, I think everybody's heard there are companies out there making therapeutic molecules out of DNA and RNA. This isn't new. There are 200 companies, believe it or not, around the world doing this. siRNA, microRNA, small RNA therapies, antisense oligos is the old one. Just make an oligonucleotide that hybridizes to a gene. It will make a double-stranded, triple-stranded molecule even, and prevent transcription. Very old idea for using nucleic acids as drugs to turn on or turn off genes. We get much more modern here with messenger RNA therapeutics. Synthesize an RNA molecule, throw it in a cell, throw it in a human, it goes to a specific cell type, gets translated into protein. Protein could be something that's missing in that person's genome. Protein could be something you want to use as a vaccination. This big area of production here. Here are just a smattering of the companies. Alnylam, Moderna, those are in Cambridge, a stone's throw from MIT. Nido Avizia, world's largest manufacturer of, GMP manufacturer of oligonucleotides, is out in Western Massachusetts, all over the world. A lot of companies making DNA and RNA therapeutics. Now here's the problem. These companies are old and well-established, and they're doing cool things. Most of the scientific basis for using those is based on the AGCT or AGCU sequence. Only in a few cases do we know that there are specific chemical modifications that prevent, or that alter the activity. Phosphorothioates, putting sulfur in the backbone of RNA and DNA, has been known for 50 years to prevent nucleases from degrading it, but that's about it. There's very little in the way of this new biology of epigenomics and epitranscriptomics that is yet built into these molecules. How do we explore this epi space in terms of putting modifications on these to alter their pharmacokinetic properties, to alter their pharmacodynamics, to make them more potent, maybe even ways to reduce toxicity by preventing transcription or translation in a cell that causes toxicity. So we really have an unmet need here in terms of defining how we can enhance these therapeutics with epigenetics. There's a biotechnology footprint here. The systems that we've discovered could be used by synthetic biologists as circuits to genetically engineer cells to do what you want them to do. We can alter translation of proteins in a way to benefit protein manufacturing, antibody production. New England Biolabs makes lots of restriction enzymes. Can we increase their production by 2 or 3-fold? They would like that, because that cuts their costs by 2 to 3-fold. New tools for industrial microbiology, where we want bacteria to make fuel. Can we put in some of these epigenetic, epitranscriptomic programs to adjust the way proteins are translated and thus the cells function in their process? The problem here is that a lot of the reagents that we need to do epigenetics and epitranscriptomics, they don't exist. There's a big unmet need in that companies who want to buy the phosphoramidites to build oligos, triphosphates to transcribe and make them in vitro, just simply the nucleosides to study, they don't exist. There are a few companies out there, Glen Research, TriLink, they just bought each other, Dharmacon, Epicipher, New England Biolabs, but each of them has a few parts and pieces, but very few have more. I'm going to put a shameless plug in for a company that I started with Liu Chuanfa and Julian Lascar at NTU, a medicinal chemist and a structural biologist, Epitoire, to make reagents, protein, nucleoside reagents for everything related to epi-everything, hence the name of the talk. All right, I'll probably go to jail for that, but it is an entrepreneurial environment here. So what are we going to do? So what are we going to do? Talked about technology that matters, talked about some new frontiers that have opened up, and here now we have opportunities to turn these into practical. We can go back and be basic scientists and study new areas. We can go make drugs, diagnostics. They're all biotechnology. What I'd like to do now is invite you to step through the door. We'll go over, have some beer and wine, and talk to see if this is exciting. All right, thank you very much.
We have a few minutes for questions, so does anyone have questions they'd like to direct?
Hi. Well, I wanted to ask if EPI makes the task of CRISPR easier or it makes the task of CRISPR difficult?
We don't know.
Okay.
This is something that my CRISPR friends at MIT and elsewhere have said, hey, you're studying these RNA modifications. What if we modify the guide RNA? And nobody's looked to see if they're modified. Does modification affect the ability of the CRISPR to target more accurately? Don't know. They are discovering how DNA modifications can affect CRISPR activity. I think there's some recent papers coming out from the big CRISPR labs. So there is an effect. Now, the RNA part of it, yeah, don't know. I'm not old enough to retire yet. I got some more grant applications to write, so we'll do one on CRISPR.
If the tRNA, the modified tRNA, if they leak out into the bloodstream, are they useful for detection of diseases?
Good question. And what's the saying? There are several heads of graduate students mounted on the wall as trophies from those that have tried to develop RNA modifications as diagnostics or as biomarkers. So this goes back to the 1970s and 80s, where someone looked in urine and looked at the molecules in urine and found nucleosides, and they were modified nucleosides in urine of cancer patients. It was mainly patients who had liquid tumors, leukemias, lymphomas, where there was a lot of white blood cells. When the white blood cells died, as they turned over rapidly, these modified nucleosides, the cells don't take them up again and use them. Regular canonical nucleosides, A, G, C, T, and U, cells will pick them up, recycle them very efficiently, but the modified ones can't do anything with them. So they get thrown out. And the physicians found that you could diagnose or follow as a biomarker the number and type of modifications, and they could only measure 2 or 3. They didn't know very much about this by simply looking at the urine of patients getting therapy. When the patients got their anti-cancer drugs and billions of white blood cells died all of a sudden, there was a huge bolus of these things that got excreted in the urine. So there was kind of a hot item for a few years until they kind of, their questions outpaced the technology. And it wasn't that predictive, it wasn't that good. But now, we've tried to look, for example, at tuberculosis infection, tried to develop a model to look to see if bacteria-specific modifications, and there are different. There's 200 of them known. Every organism's got about 50. All organisms share maybe 70%, but then every single organism, virus, bacteria, parasite, human, have about 30% that are unique and different. And some organisms have very unique ones. So if you have an infection with TB, can I look again in urine and see if there are any unique nucleosides that are getting peed out and use that as a very cheap marker? The problem is the microbiome. Your thousands of different cells. bacteria in the gut and on the skin kind of complicate this. So I'm not too confident that we're going to be able to use modified nucleosides as biomarkers. But what we found is that we're now doing studies in humans and rats, and there's a big program in the US called the Drug Matrix Program. They took rats and exposed them to 2 doses, high and low, of 600 different chemicals. I mean, bleach from the kitchen, statin, drugs, everything they could think of. And they followed the animals for 6 hours and 24 hours. They sacrificed the animals, took all the tissues out. They did microarrays for all the RNAs in all the tissues. They did blood work. So you have this huge database of animal responses to chemicals. Well, you can call them up and say, I'd like to get some liver. And you sign a materials transfer agreement. We got liver samples for about 20 different chemicals. And we extracted the tRNA, did the modification analysis, and sure enough, every single chemical did a completely different profile of modifications in the rats. So can we, for example, take whole tRNA out, just extract total tRNA out of blood and profile it and look to see, can we tell what stress you're under? Do you have a disease condition that alters modifications in a big organ like the liver? So maybe we can have a biomarker signature of tRNA modifications, messenger RNA modifications that becomes diagnostic or tells us information about that. Yeah, another grant application. Yeah.
Any other questions for Peter?
Very interesting idea about the diagnosis. Technical question, is there any connection between nanopore technology and tRNAs?
Yes. Many people are trying to now identify modifications using that nanopore single molecule technology to run it through the nanopore, and you get an unusual signal for different modifications. A little pause in the process. I don't fully understand the physics of the technology, but many people, a lot of papers coming out, sort of being able to map and identify modifications in RNA that way. Very, very crude at this point, but potentially with proper tuning, using the right pore, you can get specific information for mapping them. I prefer to use the established Pacific Biosciences SMRT sequencers to put the DNA in, and that's how we've discovered 4 or 5 new DNA modifications in bacteriophage, in bacteria, some really strange ones. That's an established one that works. The nanopore's coming, and it's a packed biosequencer, $700,000 or $800,000, and a nanopore is $1,000 for one of the chips. Yeah, USB stick. It's simple. Yeah, so if we could get that, I'd love it.
Thank you for the talk. I have a question. Do you see the quantum computing would be a useful tool for this genomic research?
Sorry, what kind of computing?
Quantum computing.
Quantum computing. Ooh, wow. You're at pay grades way above me. But what I used to, I'm a biochemist. I'm an MD. I'm a biochemist. I used to measure DNA damaged products one at a time, bench scientist all the way. I have transitioned to be, and there's some people from the research group here who will tell you, computational tools are just as important for us now as any wet bench experiment. So multivariate statistics and informatics and blast searches of genomes. Everybody's got to be a computer whiz and a bench whiz in terms of doing science. Now, artificial intelligence and quantum computing, wow, it's got to have an influence on where we go with trying to mine 10 gigantic data sets and come up with huge predictive power. But, you know, somebody else has got to do that. Yeah.
It's okay. These data sets, what type of format is it?
Oh, well, here comes one of the problems with modern data. array data is in one format, mass spec data is in another format, genome data is in another format, and are you using genes that are predicted from protein? Are you using protein sequences that are predicted from genes? Where your data source comes from? You've got to be pretty sophisticated to distinguish, and then to be able to take them all, put them in a form. If you want to do multivariate statistics on protein data, mRNA data, metabolites, you've got to massage the data to get it all into a scope and a format that'll fit into a multivariate statistics program. So it's all over the place. I wish it were all easy, because I'm getting old and I'm not learning as fast as I used to on how to manage all of that different formats, sources.
Any other questions? One last question? If not, I'm sure Peter will be available for additional discussions after wine. And Peter, thank you very much.
You're welcome. Thank you.
Now before we can actually break for our networking event, Daria will be sharing a little bit about a competition that MTech Asia is doing. And if you're under 35, or if you know somebody under 35, it's important for you to listen. Daria?
Thank you, Howard. Hi, everyone. My name is Daria, and I work with MIT Technology Review on their MTech events and on their Innovators Under 35 competition in Asia Pacific. So you all had one of these flyers. So this is what I'm going to talk about for the next 5 minutes, just 5 minutes. So has anybody heard of MIT Technology Review before? Raise your hand. All right. What about Innovators Under 35? All right, not bad. For those who did not raise their hand, MIT Technology Review is the publication arm of MIT, the Massachusetts Institute of Technology. It's a small university in the United States. So Tech Review was started in 1899 and is the world's oldest and most respected technology publication. In 1999, in conjunction with their funding, with their 100-year anniversary, the editors of Technology Review, and at some point the slides will come up, the editors of Technology Review decided to start publishing a list each year of young innovators under the age of 35. And that's where our story begins. So the list spans all technology fields, from material science to biomedicine, energy, transport, hardware, software, you name it. But what we want to recognize, it's not just the development of new technologies for the sake of it. We're looking for technology that matters. We're looking for the development of new technologies or the creative application of existing technologies to solve big problems. So throughout the years, some of the innovators under 35 that were recognized by MIT Technology Review have gone on to be incredibly successful. So I'm talking about people like, that's them, Larry Page and Sergey Brin, the co-founders of Google, or Mark Zuckerberg from Facebook, Helen Greiner, the co-founder of iRobot. I'm sure you've heard these names before. So they were all innovators under 35. They were all recognized by Tech Review for their work. But you know, another thing all of them had in common? They were all based in the United States when Tech Review honored them. So throughout the years, we realized that we were just scratching the surface, that we needed to bring the competition around the world so that we could recognize innovators not only in the United States, but also in South America, in Africa, Europe, and since 2014 in Asia Pacific. And I had a short video to show you, but I'll just skip that and get to the real part of it, which is go to the website mtechasia.com/innovators under35 and submit your nomination. So this is an incredible opportunity for young innovators from the region to be recognized internationally and join a global community of innovators that counts some of the most famous names in technology. So the nomination portal is open until the 7th of September. You can nominate yourself, you can nominate a friend, a colleague, the founder of a startup you've invested in, a postdoctoral researcher, but do so by the deadline, by the 7th of September. With this, I'm done. So let's eat. Thank you.
Thank you, Daria. And we're ashamed your video can't appear. But so again, welcome everyone, and we're breaking now for the wine and for the networking. And please, spend time, talk to each other, find out about what you're doing. There are entrepreneurs here, there are scientists here, there's investors here. So please share with each other, and let's see if we can build some of those companies that Peter was challenging us to walk through the door. So please enjoy yourself. Thank you.